All right. So yes, I appreciate the vote of confidence. I'm not yet a PhD, but I do defend in December, so soon to be. One of the main things that I really want, thank you, to kind of get across here is interest in the cyber biospace. I feel like I've pretty much stumbled upon it, but I feel like it's definitely a great need for so many different reasons, and not enough people have eyes on the space. So hopefully it piques your interest. So you probably saw just a moment ago a picture of the Twilight Zone. In this particular episode, a man was so focused on reading that he missed the life that was going on around him. Long story short, he was in a bathroom, I believe, if I haven't seen an episode in a while, and the world ended as he knew it. And at the end of the day, to make the long story short, with him reading his book, his glasses broke, and so now he's in a world that doesn't exist anymore, and he now has no way to read the books that he so much put his time into. And so I feel like right now we're at a point that we're kind of experiencing a Twilight Zone type feel here in the world, and we're paying attention to the things, to me, that don't necessarily matter at the end of the day. And that I hope that with the research that I'm doing, that I can kind of bring attention to what really matters and kind of get us on the same page. And we've already experienced some of this with COVID-19. The world shutting down by a little small virus, and it impacted not just, regions, it impacted the world, and it still has its residual effects. I know for me, I'm still having family members impacted by it, and even my own. And so with that being said, like, there's so much that can happen in such a little bit of time. Now, I started my dissertation research after watching a Friday movie. So how many of you have actually ever seen Friday? Okay, good. So you'll see a lot of images from Friday. So this particular moment, the dad is telling his son, like, how did you get fired on your day off? And it happened to be Friday. So with that being said, now with the world going in such a crazy place, you're starting to see a lot more people get fired on their day off, whether it means that somebody was stealing boxes or, just like the couple of talks that we're talking before, because, hey, they decided to lay off after an acquisition. You get disgruntled employees, or you just get people who have a lot of time on their hands, and now they want to start exploring. Something to consider. And this is an image from The Net, another one of my favorite movies. And so this particular main character, she is, I guess you could say, a white hat hacker, but she looks at different types of bugs, worms, etc., and kind of try to figures out their origins. And so in this particular scene, she comes across what's called Mozart's Ghost. It's a small script that ends up really having a big impact on her life. And so I know that we can kind of think about code in a sense of just like programming languages, but I want you to also consider code in a sense of protein sequence and how such a small thing can really change lives. So I'm not a biochemist, I'm not a structural biologist, but I do have a lot of experience from a traditional cybersecurity standpoint, healthcare, software engineering, a lot of interoperability within the healthcare space and even outside of it. So a lot of the embedded systems work that I think Dr. Ian was talking about, I do have experience with, and then machine learning. But I do computationally study proteins, so that's where I come into play. And I've had a lot of people who have tried to stop me in my tracks because I do not have that, I guess, specialty background. And I feel like for me, being someone who does not come from a traditional background, I think is worthwhile because I get to see some of the cracks that may not be seen otherwise. And so I'm saying that to you because I hope that you might feel the same eventually and see that you may not need to have say, a traditional biology or chemistry background to study or even just investigate what could potentially happen. And then on top of that, like, I've done a lot of damage just by Google searching alone. I actually kind of like got into just looking into the ins and outs of COVID-19 just by Googling it. And I've caught some people doing some pretty nasty things just by a Google search. You can Google whatever thing you're interested in and click on some links and you can pull the thread from there. And with that being said, I've actually proved right a lot of things that could be done computationally with proteins just by showing people like, hey, you do this query in Google and it'll show you exactly what to do from a protein standpoint. In addition to that, talking about proteins, so the first thing that I really want you to get out of this is like the value of proteins. Proteins in general, like we want to understand their production rates. We want to understand their function. The reason is that a lot of the processes that are out there that seem to be known have not been fully vetted within a wet lab. A lot of it's just theory. So you see a lot of research papers go into, I guess, many different rabbit holes about what potential proteins can do and how they function, but they may not have been explicitly tested over and over again. And because biology changes sometimes, that also has implications there. So with that being said, there's a lot of unknowns that we have not classified yet in regards to proteins, even just in one human body, which could change in a minute. In addition to that, proteins are interesting to me because they're actually the workers. So DNA is more so like a blueprint of what can be done in a body, etc. RNA is kind of like, I would say, customization. But when you really get down to it, proteins are more difficult because they're harder to control. And so, again, you see a lot of research that's more focused on the DNA and RNA side because you can have some bit of foundation there, whereas proteins can kind of change how they perform. In terms of protein function prediction, so learning about these proteins and what they can do, how they perform, gives us insights in ways of generating new medicines, adding new treatments for a particular disease that might not be has a cure or anything like that right now. And you can also use modified proteins for helping out with environmental things like cleaning up waste, or if you are in the cosmetology field, you might know that hair, skin, and nails are also made of proteins, and so you might have ways to improve your body and your health as well. But there is a war going on. There's the good, the bad, and the ugly. And so you have the good sides where you have things like their antibodies and enzymes that help with fighting off viruses and bacteria. You have enzymes that also help build and repair parts of your body. And then, of course, hormones that help with function. But then you have some viruses that are essentially those that kind of seek out what can be done in your body. And they also trick your cells into letting them in to do more damage. You have mutations that happen, like cancer. And then you also have misfolded proteins, which can cause like things like Alzheimer's or other functions or damage in the body as well. So proteins themselves are not inherently petty or malicious, but they can be dangerous if you don't know how to work with them. This is one of the biggest things that I've learned. For the most part, uh, as you've probably learned in your basic biology class, it is the foundation of life. However, if you change one thing like the pH or say inflammation in the setting that it's in, maybe even the amount of oxygen or CO2 coming into the space, it can change again its behavior. And then you have things like COVID-19, viruses that are not necessarily proteins themselves, but they do use them for building their structure, for allowing you or allowing them to get inside of your cells, etc. That you have to also consider that it's not just a bigger macro use cases that proteins are applied, but also on a micro level that they can have an impact. So, cyberbiosecurity. So, what exactly do I mean by that? So, some of you may have seen like 24/7 socks where you have live feeds going on. You have technicians who are looking, trying to find out what's going on, not just on the network, but also in current events or in the local region to kind of get a heads up on what might be coming. Right now, we don't necessarily have that space from a bio standpoint, but we do have dashboards that say, like organizations like the CDC, that like post, like for this particular dashboard, weekly percentages of positive tests for a particular respiratory virus that might be shown. Even though this is a good thing, this is something that somebody can kind of put in the back of their pocket and use for other things later. And then again, on a world wide scale, there's also dashboards that are also publicly available that are used to kind of track what types of things might be happening throughout the world and then on a national level. There are frameworks out there that kind of talk about how cyber and bio kind of cross over. MITRE is one that has really stuck their thumb in the space. It looks pretty similar, but you might see some new additions such as like automation in hacking, genetic treatment, so anything that's personalized to a person or a group of people. And then pathogen tracking. So you might see like things like the COVID surveillance that kind of comes out as well as part of a cyber bio life cycle. But with that being said, this is something that you would like to, one, keep an eye on, but also track along the way. So these vulnerabilities are not just something that just come once, they also morph over time. And that time can be as little as 10 seconds. Another big thing that I wanted to note about proteins in general and just the cyber biospace is that everybody is different. And when I mean body, I mean literally your bodies are different. You got different blood types, different levels of melanin, etc., that can impact how proteins perform and impact even how treatments may be consumed in your body, etc. And there has been projections from many different scientists right now saying that there are going to be many different superbugs that come to play. And I feel like, again, this is my personal thought, that a lot of the technology that has come across in the past few years are going to play a big part in more superbugs coming along and messing with the population. So now that AI has come into the chat, you have seen advances in the structural biology space, such as AlphaFold. This is just one call out. This is a tool that allows you or scientists to be able to predict the 3D structure of a protein within minutes or even seconds in some regard. So this is a process that traditionally takes, weeks, months, or even years. People have even built their whole dissertations or careers off trying to figure out the predicted 3D structure of a protein, even in lab form. So the fact that this is able to happen in such a short amount of time accelerates what can be done in such a short amount of time as well. And then you have different transformers that have come across. You probably have seen the paper or read it talking about attention is all you need. It's essentially an added layer of attention headers that allows inputs to be processed in parallel, which I'll talk about in a little bit. Before that, there was another neural network called recurrent neural network that allowed you to process things in a much slower temperament. So one word at a time, definitely a lot slower. And then it's harder to get things out as fast as you would like it. So you see older trap bots kind of follow this standpoint. But the transformer, you can kind of take in lots of text or images or whatever input you like, and then process that much faster and get big bodies of information out of it. So that's how you get your GPTs, BERTs, etc. So these new architectures have come into play and have been applied to making new treatments. So I know a couple of people had asked questions about applying AI in other contexts, but it has been in this space, and it has also been part of some of the building of the COVID-19 vaccine, over the past few years as well. That's not just the only space, but the point is that AI has been used for good, but unfortunately has also caused some headache in some areas where there have been proteins that have been used in the lab or generated by AI that have already, been tested on either animals or humans that may have caused some pretty significant outbreaks. To me, these are things that are usually kind of repressed in media, but they do exist. And then again, if you've seen Friday, you also have little Chris's that run around, right? You have the people who have been fired on their day off and they have nothing else to do. So they go around kicking trash cans, seeing what they can do, who can they make mad, and it's hard to keep up with them. And I think one of the previous speakers who had shared before had said that, now that these generative AI models are now out of the bag for pretty much most people's accessibility, it is harder to keep up from a cyber security standpoint. And I don't see us catching up anytime soon, to be honest. But we can take our next step forward to do something about it. So in terms of protein function and how AI is applied in this space, there's two models that I wanted to call out here. Originally, I wanted to share a whole gamut of it, but I felt like that might not be responsible. But there's two that I wanted to talk about: ESM, which is the evolutionary scale model, and then PROT-GPT-2. ESM is an encoder, essentially is used for a lot of classification tasks or let's just say a lot of discriminatory tasks or even explanations, where on the decoder side, think of it as just more of a model that helps you generate something new. So in this case, generating new or novel protein sequences. So these models are pretty freely accessible. Most of them are on Hugging Face, or you can just find them freely on GitHub, etc. Really, the steps are easy if you have a compute for the most part. Even if you don't, you can probably go through a notebook or some other pathway to get it working. Get the required libraries installed and start the run. And for this particular model, all you really need is to provide a seed. And the seed is just a set of amino acids that you give it, and it'll generate new patterns within the sequence for use. And then I've even tested out the model several times, and you can give it nothing and it'll also generate a new sequence. But the gag is here is like, just like Miss Parker, you know, she might look good right now, but you don't know she has a husband at home, right? And you don't know what can happen from that. So with that being said, you need a way to understand what function these models are generating. And so this particular ESM model allows you to kind of get some idea of what functions it might have. Is it an enzyme? Is it not an enzyme? Could it be used for therapeutic properties? You don't know until you kind of do a bit more research. So ESM kind of takes you out of the web lab for a little bit. And so if you have a large database of AI generated sequences, you can literally run it through this model and kind of get some hits on what could potentially work. And so with that being said, I took that same sequence from the previous slide, put it through ESM, and it predicted it as an enzyme. And it tells you more information that you need that you can literally just copy and paste in, say, Uniprot, which is a public database, and see what information kind of flows from there. Other tasks that ESM could be used, you can look at what type of mutation effects it might have. You can look at what type of binding sites. So sometimes you might not even be looking at just proteins alone. You're looking at their receptors as well, just to see what type of things could happen from there. So again, from Friday, sometimes you might think that you're tripping when you're looking at these proteins and like, hey, I don't know if this is actually going to do what it says it's going to do. Or in some situations you might be tripping and is really Debbie on your lap. Or you might get somebody that looks like a Janet Jackson, but you know, comes out like Freddie Jackson. So what I mean by that is you might get a sequence that says that it has an enzyme property, but might end up having other residual effects that might be impacting on a human. So the several governments, including the US, have stated that AI tools could be a good use to defend against these AI-based protein engineering tools. But the thing is they also admit that there aren't many mitigations or protections in place to defend against them. And then there's a race. I mean, there's other nations who have also been talking about this, but no one has really put out any like regulation or restrictions on how, you know, these things should be built or how they should be applied. And then you have a race with other nations who are, you know, kind of weaponizing these tools, and they might even say that they're using it in drug development. But we know, like toxins, for instance, this is a toxin that comes from sea snails. You know, this is something that's used for, say, neural-based treatments that could, one, yes, it can be used for treatment, but on the other side of it, it can also be used as a weapon of mass destruction. So with that being said, guardrails, from the research that I've done so far, 100% of the models do not have any guardrails in place. And what I mean by that, there's no flag to say, hey, you just created a potentially malicious enzyme, or you may have created another toxin that could impact this demographic. Or like, hey, you should probably wait 10 seconds before you generate another sequence. None of that is in play, or even in the folding standpoint as well. So there is an urgent need for it. And for me, one of the things that really makes me kind of think about what needs to be done in the space is this scene from Friday. And basically, just like Debo, he's not fighting fair. Cyber criminals are not fighting fair. And so with that being said, they can take something as small and simple as this neurotoxin that can selectively block neurotransmitters and turn this into something that spreads, make an aerosol spray from it. You could set this as something that is labeled as a non-toxin and put it into some everyday use item. And with that being said, a lot of the different times these AI models, again, even if they're independently used by another model, like I think another speaker was saying, they could also accidentally be used or generated as well. So even if there isn't a human in the loop, sometimes it can happen as well. So with that being said, I had a big talk with Big Worm and basically told me I wasn't applying myself enough my first year of my PhD. So with that being said, I decided to go into this space and start looking into these models and really kind of grinding and seeing, okay, I want to evaluate what dual uses could be had, what good can come from them, and also what bad. So for instance, I'm right now building a model called Craig. It's essentially based on a framework, but it's also a model as well. That essentially flags some of the risks that might come from these models. So my hope is to have this as an add-on or maybe a wrapper for some of these models so that you can actually flag some of these things that are coming from it. In addition to that, I'm creating another model called Debo. And the goal there is to kind of discriminate where are the origins of these models. So I'm taking in many different models, generating tons of different protein sequences or even their 3D predictions, and then seeing if I can reverse engineer essentially and see where do they come from, so that if someone does create something crazy and they put it out, we have a way to at least kind of identify and what type of model did it come from, and that at least gives a step ahead of catching up with Lil Chris. And one of the things I wanted to mention is that I know a lot of times in conferences like that we talk a lot about like what type of hat we might have. Honestly, it really doesn't matter at the end of the day because we're all in the same race. And I wanted to say that at one point I thought of myself as gray hat just because I needed to do some kind of like edgy things in order to kind of get an understanding of the space and see what bad could be done. But I've already caught people, and people have not been happy because I'm a person that Googles and they feel like I'm not smart enough, and I've also helped organizations find other people who are doing this internally and externally. And it was because they thought nobody was watching. And also, with that being said, my hope is to eventually have some type of tool that can be deployed in space to help with responding to these incidents. And yeah, I want to say, and the gag is, it doesn't matter who you are, what your background is, you can get into this space, you can help out from a cyber biosecurity standpoint, and you can definitely try out these models and see what good can come of it, what bad can come of it, and join the research race or just the hat race and trying to prevent this from happening. So if you want to connect with me, feel free to. I do have some papers that have been called out here. And then I do have a way that you can try out these models like via notebook. You will have to go through another layer because I want to track who's doing what with it. Sorry, but I don't want nobody coming after me. But yeah, feel free to use the QR codes. Happy to answer any questions you got. Thank you. Sorry for my voice.
thank you so much. This is something that I think about a lot, and I did not expect to hear this. I just thank you, God,
Aw.
thank you. You're a genius, and something that I think about a lot. And bioengineered prions make me want to die. So thank you.
Well, definitely connect.
I piggyback off of the thank you as well. Thank you for being here for our village community as well.
Absolutely. So I have a question. As you alluded to in your talk, it's actually quite difficult to test proteins in real time in experimental models. But in these, sort of synthetic tools that we have now, we can create sequences and modify sequences in silico. But it's much more difficult to potentially produce those proteins and actually test them. So you may find that people are generating these malicious sequences potentially, but how concerned are you that they're actually able to translate that into some sort of real world bioweapon potentially?
Yes, so I've created, let's say, 40 assays that are wet lab based. So from an E. coli standpoint, and then several human tests to validate what has been generated in silico to see how well it translates from the lab, essentially. I've gotten to the point where I've gotten 90% accuracy on what has been created. And I think that that information is going to come out not necessarily by me, but by other people eventually. So from what I understand the question, you're saying that how are bad actors or how are scientists generating? Okay. So there's decades of research that have been put into like how proteins do function and what mutations do kind of come from an evolutionary standpoint. You can see this from, again, these public databases where people have taken the time to annotate every single portion of a protein where they could. With that being said, before AI became a big hubbubaloo, there have been, computational models that were used prior to help with predicting function. So with that being said, over time, they would use these models and then test it in the lab, as she was mentioning, and then validating what they see. So the mutations themselves, I don't think is a mystery because they're bound to happen. But in terms of what impact they might have is stuff that needs to be at least tested in the lab at some point. Yes. Yes.
Yeah, thank you for this talk. This is an exciting area. I've currently been in cybersecurity for, time flies, like 15 years now. But prior to that, my master's degree was in like biochemistry, genomics, and bioinformatics. So it's just great to see like the kind of conversion of this space. What I'd like to know is you kind of touched on at the end where people can get involved. So something I want to do is like get back into the space. So can you talk about like at the different levels or backgrounds where you think people can contribute? Like you touched on it, but if you can give more information.
I love that. Yes.
Awesome.
So one of the things that I'm trying to do is I guess I'll answer it two ways. One, I'm hoping to get you have like standards organizations and things like that. I'm trying to get at least some type of working group together. It's proven difficult given that my studies take a lot of time. But hopefully you'll see something come of it soon. It may not be official, but my hope is that. The first stance is if you are in a developer standpoint, I would say there's several I would call mom and pop models that have been published that do not have any type of control in place. So going out on GitHub and looking at these models, I can provide you a list of the top 10 that need attention today, if you'd like. And go in and start contributing in a sense of flagging where you see transitions between generation processes, looking into the evaluation of the models is really important, and even the training data that they're inputting. From a national database standpoint, there are a couple of holes. Well, there's a lot of holes in these databases. Some of them are some proteins or peptides are really well studied, and some just have generic information hey, this is the sequence name. It might have a name unknown, and that's about it because nobody has had the resources to study them. So even going through and computationally running it through tools that do exist and just applying it to those public databases would be helpful. Yeah. Hi.
Considering some interactions between proteins and substrates, because some proteins works as an enzymes, right?
Yes.
And the enzymes work with a substrate.
Mm-hmm.
So there is tools in this model that consider these interactions,
Yes.
or it's just the if the protein is functional or not.
Okay, if I understood your question, well, yes. So you're saying like, is it also considering like the scaffolding or like other pieces other than just the protein sequence?
There is a tool that in the model that consider the interaction
Oh,
between, for example, a protein that is an enzyme with a substrate.
yes, there are. Yes, there are. I don't know the names of them right off the top of my head, but there's a tool called NeuroSnap. If you Google it, it has like basically a cloud-based repository of different models, and there's at least 3 of them there that you can actually kind of see how the interaction happens with something, or like you kind of put in another sequence or another protein, whether it be in its 3D form or its sequence form, and see how well it might interact. Again, this is something that you would still have to test in lab, but it does a good, really good job of doing it. And I feel like more models like that are going to come out as we progress in this space, for sure. Yes, no problem.
Okay. I'll do this one, and then Xavier will be last.
Thank you again for being here. I really appreciate the information.
Yes.
So my question is for us lay people, how do we become people who Google? What do we need to look out for so we can say we're people who Google as well?
Oh, yeah, that's a great question. So you're saying, okay, I would say get familiar with one, what an amino acid is and how they come together to make proteins or peptides. Get familiar with some of the public databases. So for instance, if there's another COVID-19 that comes around, get an understanding of what that actually looks like from a text perspective. So now you kind of have an image. Start putting it into the public database and seeing what does it look like in this 3D form. So when you see on the news like a picture of it, is that accurate? Is it not accurate? What types of effects they might have might not be what you hear from, say, family members. Like for instance, I was on a team that essentially helped discover COVID feet and COVID hands, or the fact that like the first 10 symptoms was saying things like, you might be red and blush in the face, but I mean, not all melanated skin show red and blush in the face. So just because you state that as a top 10, they might be missed in, say, the hospital if they go in for an appointment or a screening. So with that being said, doing some validation, even if it's just the Google search, to kind of validate what you see and hear from the news. And if there isn't some validation, start posting that online. It doesn't really matter where you post it, because somebody who's interested or doing this work is going to find it, right? So it can be Reddit, it can be Twitter, it can be wherever you want. And then somebody is going to start picking that up when they're looking through. Yeah.
Yes, I want to thank you again for your presentation. It's a breath of fresh air because I don't believe enough people are aware of cyberbiosecurity, and not enough people understand the gravity of it in terms of all the implications that are in store. With respect to the questions, or the mention of wanting to start a working group, yeah, I'd be down to assist.
Oh, heck yes.
And also have a colleague who would be willing to as well. And yeah, just let us know. But also, I want to share this time. I want to share this comment space also with the other gentleman, because I can talk to you after.
Okay. Yes. Let's do it. Thank you.
Hi. Thanks for a great talk. I had a question regarding kind of towards the beginning of the presentation. You broke into a field that your background didn't quite specialize in, and besides that mindset of well, I'm gonna do it anyway, do you have any other wisdom to impart on going about that? Like, for example, did you also maybe need some buy-in from your the head of your committee or your doctor or whatever?
That's a really good
Any other-
question. Yes and no, and I'm saying this safely as I possibly can. So, my first, or how do I say this? I got a hold of these tools. So, my research is actually going to be focused on a standard called FIRE, completely different from what my dissertation is now. And I was just looking up different ways to generate synthetic data. And I just happened to come across like models that generate synthetic proteins because I'm just looking at every angle. At the time, unfortunately, I didn't really have a structural biologist or a chemist like right by my side, but I just started generating proteins. So when I found something that I thought was potentially dangerous, that's when I started going to folks and saying, hey, like from this model or these models, I created this. Can you give me your take on it? And so that's where I was starting to get expertise. Now, starting from vanilla, I do recommend having somebody there because within 30 minutes, I created a pretty nasty version of HIV. And I was trying to go through the proper channels, but I didn't know where to go, which is why this working group would be really good. So I just started Googling people, and luckily I found somebody who helps run Uniprod, to make the long story short, and they were able to help guide me with what to do with that information. So yes and no, and my advisor, he's in complete alignment with me doing this work. I do have a couple of chemists and physicists and structural biologists who do help me out because mostly the lab stuff I have not necessarily done just because I have to have a drawing line with my research scope, but I do like help set up the essays and kind of pour in a lot of my perspective in the design process so that it kind of mimics what it looks like on the computation side. So, yeah. You're welcome. Yes. Thank you. That's a really good question. Honestly, if I'm if I'm being completely transparent, I don't think that we're going to catch up as fast as we need to. There has been some guidance, say from like the FDA, where they're talking about new biologics, and this was like recently, like February 2025, where they've given guidance on like biologics generated via AI. But they don't necessarily go into the biosecurity standpoint. Do you get what I'm saying? So it's just like, okay, they start talking about how a model might be trained and, maybe the residual use, but nothing to kind of block flag or kind of show users what type of damage could be done. And in terms of a more, I guess, global or even generalized framework for this, I think it will take a lot of players who are on one accord to really adopt it. And I feel like for me, the reason why I'm still trying to go up this uphill is because of the company Stride. They started off with a couple of lines of code, and I felt like they really changed the game by just starting with developers first, getting them to be enticed. And so with that being said, why not go to cybersecurity experts, or at least people who are interested first? They see the issue. They might do something about it. I can't do it alone, obviously. So that's my stance. Yeah. Sorry.
All right. Okay, this will be the last question. But thank you all for being very involved in everything.
So like the models you're talking about adding guide rails to, is that like the open source models, or is that like the actual providers that people are using? Like the
Yeah, but ESM is a meta model, and it is open source. GPT 2 is also open source. It's from like, I forget, some academic organization, but there are a lot more out there. I just wanted to refrain from naming them.
So like if it's an open source, like even if you add a guide rail, like a malicious actor, like it's already out there, isn't it? Like
Sorry, repeat that. I didn't understand the question.
Like if the model's already open sourced, like adding a guide rail doesn't stop a like malicious actor, right? Like they could build their own.
Yeah, no, I mean, I'm not saying that my solution is the solution. It's just a solution for people who want to do right, right? Just like there aren't any regulations for AI models in general, they're already out the bag. So unless somebody really wants to make, or at least take a stance and say, hey, I'm doing this ethically, then they'll probably add something like it to it. One of the biggest reasons why I do want to have some type of working group is to figure out how can we retroactively do this. I don't know what that would look like. Hey, a lot of the models weren't even technically quote unquote AI based. Again, a lot of them were just basic linear regression models, used for making mutations. So the cat has definitely been out the bat with that one, but I do think it's still worth a try. So. Yeah, thank you.